Welcome everyone to Wisconsin Sea Grant sponsored Students Ask Scientists presentation. Today we're featuring Anna Hill from Purdue University on her presentation, Should I Stay or Should I Go? These presentations are done in cooperation with the Center for Great Lakes Literacy, which is not a physical center, like a building. Rather, it's a group of educators across the Great Lakes basins that promote a variety of educational opportunities for both students and educators. At the end of the presentation, we'll give you a link to the Center for Great Lakes Literacy website where you can find a variety of resources related to the Great Lakes on many different types of topics. After the presentation, we will transcribe the recording and eventually it will be posted to that website. This takes a significant amount of time. So if you're hoping as an educator to show it to your students who may be absent from class today, it will not be available tomorrow for that purpose, but hopefully in a week or two, you can join us for that. We do have an opportunity for you to ask questions. Please use the Q&A function of Zoom rather than the chat function for that. And our first question for you is if you would please put in the Q&A the city and state where you are currently located and the number of individuals that you have in the room with you. That helps us to get an idea of who our audience was today for our reporting purposes, and we thank you so much for that. And so without further ado, I'm going to turn it over to Anna for her presentation, Should I Stay or Should I Go? Thank you.
Hi, thank you, Ginny, for that wonderful introduction. And I wanted to say thank you to Ginny and also Jamie for allowing me to kind of connect with some students and talk about the research that I have spent the past 2 years of my life working on. So as Ginny said, today I'm going to be talking about yellow perch. So should I stay or should I go? I'll be talking about yellow perch movement in eastern Lake Michigan and its connected John River Mouth Lakes. That is not a yellow perch picture on the screen that you see. I couldn't find one of me holding one that was appropriate. So this is what we're going to have to work with. So before I get into kind of the meat of this presentation, I wanted to introduce myself. So I'm originally from Chicago. I went to college at the University of Delaware to pursue a bachelor in science in marine science. And my concentration is in marine biology, but I kind of got a really well-rounded education in chemistry and oceanography and biology and all these kinds of different marine-based things. I then kind of took 2 years off of schooling and I went and got jobs. So I worked as an aquatic technician for 2 years in two different agencies and then decided that, hey, I probably should go to graduate school. And that's where I'm at today. So I'm a master's student at Purdue University. I work with Tomas, Höök, and Paris Collingsworth on a number of different projects. This is just one of them. And just some fun facts about me, because why not? I love knitting. I love it so much. I also can scuba dive. I've been scuba diving since I was 13. And I play a couple instruments, just a few. And my favorite fish is a lagoon triggerfish. However, we will not be talking about that today. We will be talking about a different species entirely. So to start this presentation, I kind of wanted to ask the group if they could use the Q&A function to answer a couple questions for me. Mostly, I just kind of wanted to know what you guys thought of when you thought of Lake Michigan. This could be literally anything. So when you think of Lake Michigan, what's kind of the first thing that pops up in your head? I know it's hard over Zoom. Niske? Jamie, I'm not quite sure I know what that is. We can talk about that later, though. Your favorite fishing spot? Awesome. That's really cool. There's a lot of obviously Lake Michigan has a ton of fish in it. And so there's lots of fishermen, lots of fishing going on in Lake Michigan. So that's actually a big one. So the research vessel with School of Freshwater Science. Yep. That's also a really important one. So just some broad-based understanding of why people use Lake Michigan and what they use them for is one of them is charter fishing. So there's a huge charter fishing industry in Lake Michigan, including salmon including lake trout and yellow perch and all different kinds of fish. There's also some recreational fisheries that a lot of people kind of indulge in, which I saw Max, you mentioned. I'm from Chicago, so when I think of Lake Michigan, I kind of always think of Chicago, and it having really good, beautiful views and great access to the lake. I also think about the eastern shoreline of Lake Michigan, so all the beaches that there are provided. Also lighthouses. I'm not sure if anyone knows, but there's a lot of lighthouses in Lake Michigan, on Lake Michigan. And then, of course, Sleeping Bear Dunes, which I did when I was a child, but can't do it now. So we as humans use Lake Michigan for a lot of different reasons, a lot of them recreational, but there's also some commercial aspects to this. And not just that, but Lake Michigan is also home to a lot of connected habitats that fish use throughout their lives. To name a few, there's a couple of tributaries, some rivers, the St. Joe River down here and the Kalamazoo River. There's bays and estuaries, which I'm sure a lot of people know about, Green Bay being one of the bigger ones. And I'm pretty sure it's one of the bigger ones in freshwater in across the world, really. And then we have Traverse Bay over here. And there's also these smaller systems called drowned rivermouth lakes. And here's just a really quick image to show you just how many there are. And they're mostly located on the eastern shore of Lake Michigan. And fish can use all these different habitats to do all different kinds of things. So this mix of habitat supports all different kinds of fish species, not just in their adult life, but also in their main stage, any stage of their life. So we can't really move forward without kind of talking about the characteristics of these habitats. So what you're going to see in the next couple of clicks is a variation of the same sort of setup on the graph. So first we're going to talk about temperature. The triangles will always be Muskegon Lake, which is one of those drowned rivermouth lakes, and the closed circles will always be Lake Michigan. So these samples were taken by a PhD student down with me here at Purdue. And all the Lake Michigan samples were collected near shore, so within the first 30, 40 miles offshore. Right off the bat, you can tell that these drowned rivermouth lakes, specifically here, Muskegon Lake, is quite warmer than Lake Michigan. That provides really important for fish because certain fish will spawn in certain temperatures and they won't spawn in other temperatures. Ideally, fish usually generally like the warmer temperatures more. Turbidity, and if you don't know what turbidity is, it just means the clarity of the lake. Jamie can talk about that because they did a little bit of that when we were on the boat together. But as you can see, it's pretty starkly different. So Lake Michigan is fairly clear. That has to do with a lot of different things, mostly its size. Also, there's these mussels that are in Lake Michigan that kind of are clearing out that water, which is kind of a whole separate issue. But you can see Muskegon Lake is pretty turbid. Turbidity offers a lot of different things. So it offers protection from predation. It offers food. Usually turbidity means there's a higher food availability. Usually fish will lay eggs in certain turbid environments. So having a very turbid lake is generally pretty good for fish. Speaking of food availability, we have zooplankton biomass on the left. And so zooplankton are just a really important key prey for fish, kind of all fish. And so you can see here that in Muskegon Lake, there's a lot more available than there are in Lake Michigan. And so when fish are thinking about where they want to head, like where they want to be, they generally want to be in areas that have a little bit more food available to them. So the two systems, like Michigan and Muskegon Lake, which I'm kind of using as a broad blanket for all the drowned rivermouth lakes, they are very variable. They're very variable in everything we just talked about, turbidity, food availability, temperature, but also in wave action and protection from like different atmospheric climates. And they offer different benefits for different species. So different species will use a combination of these characteristics to do different things in their life. Now, here's another question for you guys, and you guys can just throw that in the Q&A function again. But what are some reasons that you think fish move? And I mean, not just move like 5 feet. I'm more thinking like moving big distances. I'll give you guys a second. Food, good. Food's a big one. Spawning, also a huge one. Mating, also good. Not enough food, temp change to lay eggs. Yeah. So these are all really great answers. Changing in currents and weather. That's a really not a lot of people know about that one, but that one's also a really big one. So for scientists like me, just very broadly, one of the reasons they move is reproduction. You might know salmon, they like to go up to rivers. Some fish will move hundreds of miles to lay eggs. A lot of this is kind of mysterious for us, and we're really interested in learning why they do these kind of things. Another reason is for food. So, as I said earlier, those drowned river mouth lakes tend to have a little bit more food than open Lake Michigan. So, we would expect fish to be migrating to these little areas to eat, which we may or may not be seeing. Seasonality, seasonality and climate, they're kind of one and the same. Some fish like warmer waters. So when I think of this, I think of like snowbirds, excuse me. So we as people, some people like to move down to Florida in the winter because it's warmer. Fish can kind of do the same thing. So fish will kind of move with the temperature of the water and then avoid predation. This one is a little bit not as common, but we do see some fish moving pretty far distances to avoid being eaten. So now that we kind of know our habitats, the characteristics, why they're moving, for me, my study is pretty heavily focused on yellow perch in Lake Michigan. So just some really quick, rough and dirty facts. They're found in all five Great Lakes. Yay. They're easy to spot. They're golden yellow body and greenish stripes. People really, really like to fish for them. I have a lot of people when I was doing the study come up to me and be like, "Where are the fish at?" Really excited and really interested in yellow perch. Their diets consist of small invertebrates and fish, and so they're usually eating little larval fish, which is just another word for baby fish. And they're very important for both local fishermen and commercial fishermen. I think they are contributing to more commercial fisheries than a lot of other species of fish. So, quick recap. Lake Michigan, not just for people, also for fish. A lot of different habitats in these lakes. These round river mouth lakes are big, the tributaries are huge, the bays are really important. These habitats offer different resources. Some habitats have more resources than others, and we want to know why fish are moving into these systems to figure out what's going on. Like what we said, why are they moving? Reproduction, food storage, forage for food, seasonality, all that kind of good stuff. And fish often move hundreds of miles for these resources for different reasons. And yellow perch are an economically important fish, not just for me, but also for lots of people who like to eat them and collect them. So good, we're done. Science is done. We've learned all we need to know. We're good. Not quite. Not quite. So now I'm not the first person to be interested in yellow perch in Lake Michigan. This study here, done in 2019 or published in 2019, was pretty much my foundational for our understanding of yellow perch. So even within a single species, not all populations use habitats the same. Some groups rely heavily on certain habitats more than others. And these conditions can reflect unique environment conditions, life history strategies, and local adaptations. So this paper by Chorek et al., which was published in 2019, we use as kind of like a baseline case study. The goal of this study was to examine how population structure of yellow perch relates to habitat connectivity between Lake Michigan and these drowned river mouth lakes. So specifically here, they looked at Muskegon Lake, which is the one that I'm also interested in. They did I'm not going to get too into the methods because it wasn't my paper and I'm not a geneticist, but they did genotype yellow perch from multiple different drowned river mouth lakes and nearshore Lake Michigan sites across seasons. Biggest finding for them, there's two genetically distinct populations of yellow perch in Lake Michigan. Now, what this means, genetically distinct, is that they are not spawning together. That's big. We need to know that. So we have two very different, genetically speaking, Lake Michigan yellow perch. We have Lake Michigan residents, and we have these drowned river mouth lake residents. And what it looks like is that there's really no moving in between, or so we thought. So what we know now is that there's a subpopulation that move into Lake Michigan, and that's what Taylor Senegal looked at. So Taylor Senegal published a paper in 2020 that looked at those two resident populations of yellow perch, trying to figure out if there was moving in between them. So they wanted to determine whether yellow perch in eastern Lake Michigan and drowned river mouth lakes represented distinct resident populations, or did they include a portion that were moving in between. They did a different approach, so they used stable isotope ratios in their otoliths, which is their ear bones, and then their muscle tissue, to trace the natal origins and recent habitat use. Their biggest finding was that they did actually find a subpopulation of that Lake Michigan residents that was moving in between these two systems. So now what we know is that there's these Lake Michigan residents, these drowned river mouth lake residents and a very small section of the Lake Michigan residents that are moving in. We call those the Lake Michigan migrants. That's great. So now we have all this information. What don't we know? This is another question for the chat. Just based off of what I've been saying for a little bit, what are some of the knowledge gaps, or what questions would you possibly have about these two populations of yellow perch in Lake Michigan and their drowned rivermouth lakes? And I'm gonna have some water. This is not a trick question either. I will take any answer. Well, if you were thinking why we have these two populations, we have a populations that are moving in between, but we don't know when. There's been some anecdotal, which means just like word of mouth. There's been some anecdotal information that yellow perch are moving in during the winter and moving out during the spring, but there hasn't really been a whole lot of hard kind of data on the timing exactly. So one of my research questions was, when are these yellow perch moving into and out of these drowned rivermouth lakes? Again, we think they're moving in late fall and they're moving out in late spring. We have no data to support that except for a couple of visual markers. Another question we kind of came up with was, we were wondering if once the yellow perch left those drowned rivermouth lakes, did they return to the same one year after year? We have no reason to believe that they wouldn't, but we also have no reason to believe that they would. And so, as, using this as kind of our baseline for our research questions, we used something called acoustic telemetry to track these fish. And what acoustic telemetry is it uses three main parts. There's a receiver, which you can see on the screen. There's a transmitter or acoustic transmitter, which sits inside the fish. And then there's, of course, the fish itself. The easiest way to explain this is to think about it as like a cell tower. So as you're driving down the road, your cell tower connects to your phone, kind of checks in with it and says, hey, I know where you are, keep moving on. Same thing happens here. So we take these tags, we put them inside our fish, and that tag emits a ping every 180 seconds. And if that ping goes off while it's next to a receiver, that receiver will just kind of take attendance and say, hey, I see you, we're good, keep moving. And this is a really great tool to passively track fish, which just means it's kind of hands off. We pull the receivers up, twice a year, offload the data, put the receivers back, and the fish can continue doing what they're doing. But we can't just place receivers in willy-nilly. We kind of have to make kind of executive decisions when it comes time to put the receivers in the lakes. So we use what's called an array. An array is just a kind of a specific way that you organize your receivers so that when a fish goes through it, you can get a very, very, very specific point in time where that fish was. So again, these receivers are specifically placed stationary, and they allow us to passively track these fish. So the trail of dots that you see coming out of that fish's butt is not poop. Those are just kind of, they're not, you don't see them in real life. But each dot kind of coincides with one detection or one ping that fish is sending to that receiver. So fish can swim around, we collect all this data, life is good. So that's basically what we did. So this is just a map of all the receivers that kind of I have my eyes on. On the top right, you're going to see Lake Charlevoix. I did not place any of those receivers, so I won't be talking too much about that. I'm going to focus on Muskegon Lake since I put all those receivers in the lake. And on the left, you can see kind of the two methods that we did. You'll see a cement puck with a white tube or white PVC pipe sitting in there. A receiver went in there, and that puck we then dropped in the lake. So all the open face circles, 1 through 8, are all the ones that are cement, and they're sitting on the bottom of the lake. The important ones for us are the black circles, so that 3, 4, 5 on that bottom right map. So 3, 4, 5 are the receivers that are sitting in the channel. And so when we're thinking about tracking fish that are moving in and out of two systems, we are really lucky that those drowned rivermouth lakes are connected to Lake Michigan via kind of a very narrow section of water. And so we put 3 receivers in the channel to kind of help track the fish as they were moving out. And we were able to deem a fish has left if it hit 453 or 345 in a in succession. And so now we have all the receivers out. We had to get to tagging. I wanted to throw in some pictures because I think this was when I was starting to get into the science. It was really cool to see kind of people doing things. So these are just more pictures of the receivers that we put out. Those cement pucks on the left, there were 8, and then we put them all out. The ones that are attached on the channel are attached to the emergency rails that are kind of on the side of the channel using this chain. And it was, I think it was December, November when we put these out. So it was very cold and very unpleasant, but fun nonetheless. So now that our receivers are out, we have to put our tags in something. So the way that you use receiver tags or acoustic tags is you surgically implant them into the yellow perch. Don't worry, the fish are fine. We have a whole host of protocols that we follow. We anesthetize them. We make sure that they're comfortable. We put them in 2 tanks to kind of make sure that they're going to be okay before we release them. And another important part about this is that you have to make sure that the tag is light enough to avoid affecting the buoyancy of that fish. Because if you put a tag in that's too heavy, the fish is going to sink. Unfortunately, it will die, and then you have no data. Another thing to think about when you're planning out these kinds of studies is the battery life of each of these tags. So each tag has a specific amount of days that it operates for, and that can kind of be a constraint when thinking about how long you want to run your study for. So for fish tagging, the fish were angled, so we used hook and line to get these fish out. They were taken out of different depths. Because we were taking these fish out of pretty deep depths, we experienced a little bit of barotrauma, which just means a fish that is pulled out of a depth that kind of changes pressure very quickly, experiences some physical trauma. We were able to deal with that and kind of put them back in. Okay. And then we did all this in the winter. So I'm pretty bundled up right there. It was very, very cold. And it was very hard to do surgery on a fish when it's that cold. So in total, we tagged 38 fish, 19 in each lake, which is great. We did lose 1 in each, but that's kind of just how the cookie crumbles with this work. Here's some more pictures of science in action. On the left, it's me doing the surgery on these fish. In the middle is on the top are the yellow perch that we collected. On the bottom, you'll see we somehow got a pike as well, which was pretty cool. And then it got cold and a little bit boring, so we were goofing around a little bit. Okay, here are my results. There's a lot going on here, so let me just kind of make sure we are all on the same page. On the right-hand side, or my right, you'll see a map. That's just the same map as before, telling you where all my receivers are. That's important because, again, we need to make sure that we're catching these fish leaving, so hitting that 345 or 543 on the way out. On the left, you'll see what's called an abacus plot. And the abacus plot is basically a way to visualize the detections. Again, those detections are just when that the tag hits the receiver. So each on this plot, you'll see a bunch of little squares. Each square represents 1 fish. On the top, you'll see tag ID, so that 33630, that's just the serial number that we use to identify the fish with who it is. So when we tagged all the fish, we wrote down all of its information, and we were able to then go back and kind of figure out who is who. On the y-axis, you're going to see receiver station. Again, the big ones here are those CHH 005, 4, and 3. So those are the ones that are on the channel. And everything above that are just the Muskegon Lake ones, 1 through 8, which you can see on the map. So right off the bat, we're going to talk a little bit about mortality. So all the ones in red, we kind of have deemed either false detections or it passed away. And the way that we were able to kind of understand that is that each dot on this map is a detection. And so you can see for that top middle one, that there's a lot of detections on 1 receiver. That's pretty unusual, as you would expect a fish to be moving around a lot more than that. So we assume that that fish has either passed away and is sitting right next to that receiver, or that's kind of all we think that happened. So those 4 that are highlighted in red, we are pretty confident have passed away, and we were not using those in our final analyses. It's hard to see, but the ones in yellow are the ones that we actually think have left. So, yay for us. Of our 19 fish, we were able to see at least four leave, which was pretty exciting for us because our sample size is fairly small. Um, Again, they left. You could see that they left because they hit those receivers in succession. And I'll kind of explain a little bit more about the timing of those fish leaving as well. But there was one fish that kind of did something interesting. So this one in blue. And I'm going to move on to the next slide, and that will help you hopefully see a little bit more of an interesting story. So, as I said before, we had four fish that left. This is kind of a blown up version of that same plot as before. So on the y-axis, you're going to see receiver station, and on the x-axis, you're going to see the timestamp. So you'll see kind of four big juts up into the data. So you'll see triangles, you'll see circles, you'll see squares, and you'll see plus marks. So as you can see, these fish are moving and leaving the system not all at the same time. They're kind of using like a pulse migration type movement, which is fairly interesting. We kind of assume that these fish just left and came back in at the same time. So this is kind of showing us that that's not happening. What I really wanted to show you though is that that transmitter or that tag that's circled in red, so all the plus marks, that fish did something that we no other fish did in my study. So my fish, that fish, I think took the snowbird mentality and kind of ran with it, where it was in Muskegon Lake, it hung out, it did its thing, it decided to leave, and it just headed straight south. And so we were able to get that data from a network called the Great Lakes Acoustic Telemetry Organization, which is just like a really big powerhouse acoustic telemetry organization that houses a bunch of data. And so we were able to understand and follow this one fish throughout its life, moving further down south. Again, this is all pretty preliminary and descriptive, and so we don't really have too much of like a why they did what they did, but it is important for science to know what is happening before we can ask why it's happening. So what does any of this mean? So far, what we know so far, again, there's two genetically different groups of yellow perch in Lake Michigan. Some of the fish from my fish, those 19 left, but they're not leaving all at the same time. So they're not doing a solitary movement all at the same time. So they were all leaving at different times. I think the earliest was January and the latest was must have been April. And so that's pretty exciting. And we didn't observe any of the fish returning from the lake that they came from, which again, we weren't really expecting them to, but we weren't expecting them not to. So we were pretty interested in why they were doing that. And then to kind of bring this out into a management lens, knowing when and where fish move can help managers like the DNR make more informed decisions about harvest timing and location, which and then impacts you guys if you are recreational fisheries, and sometimes also the commercial fisheries. And so an improved understanding of movement patterns can lead to regulations that make better protect key habitats and the fish that use those habitats. And with that, I'd like to thank quite literally everyone that's ever helped me with this project. So all the fish in Charlevoix were tagged by Ben Tershak from Michigan DNR. I have named some of the people that helped me angle these fish. Also, the members of the lab that I'm part of, the Höök lab, who were out there in the cold in December and November, tagging all these fish and collecting them. And then these are just some of the agencies that have kind of helped me put this all together. And with that, I have some more action shots of me and fish and boat. And I will take questions.
So I'm going to jump in for a minute here and just help the audience get some information about how to facilitate the questions and answers in case they haven't participated with us before. But first and foremost, Anna, thank you so much for putting this presentation together, for your willingness to share with students across the Great Lakes Basin. We had opened it up to folks not just on Lake Michigan, but other of the Great Lakes as well, because perhaps similar things are occurring in the other Great Lakes basins. For those of you who may have joined a little late, we did ask at the very beginning of the presentation for this to happen. So if you have not already done so, could you please put the city and state where you currently are, and how many individuals are in the room with you listening to the presentation. That helps us to take kind of a little better idea of where folks are located and the number of individuals that were participating today. The webinar format is a little bit different than perhaps you're used to with Zoom. So there is a separate Q&A. It looks like most of you have already figured that out. So do please keep putting questions into that format for Anna to be able to answer. Anna, if you haven't managed this before, there's three different little tabs. You should be able to see a button that says open.
Yeah,
So as you've been talking, Anne and I have been answering questions that have come in to the best of our ability, and I have kind of put those into the answered column. And then what we want to do is address the questions that are still open. So
gotcha.
if you could help out for the recording, and before answering the question, if you could just read the question and then answer it, that would be lovely. And in the
Absolutely.
meantime, please keep adding more because we've got about a half an hour to try to address some of the things that you might be wondering about. I'm
Yeah.
going to turn my camera off and turn my audio off again and turn it back to you, Anna.
Okay. So I'm just gonna kind of start at the top and work my way down. You guys are more than welcome to throw questions in there as you go. So Jamie asked, what is the actual difference in their DNA? Super cool question. Not a geneticist. There was a difference between the satellites, something with satellites. I can send you the paper if you want to read it. But I just kind of briefly went over it. There's lots of cool figures in that paper as well that I can kind of send to you. And it was a really interesting paper. And again, it was one of those foundational papers for this kind of study. And there's been a lot of work since then to kind of build a genetic sequence that for yellow perch in Lake Michigan that other people can then use with for like forwarding on research. So thank you for that question. Max asks, how do they survive under the ice in the winter? Super cool question. So yeah, a lot of these systems, because they're so small and shallow, they do freeze over, which is great for all those ice fishermen out there. But the fish will survive in kind of the way you think. So these lakes, all the bottoms are very different. Usually there's like one spot that's pretty low. And these fish will just kind of either hide in there or again, there's vegetation, so they can kind of swim between the vegetative all the vegetation to survive that way. Unfortunately, some don't survive. They do get angled. But at least for me, I don't know if I know of fish that freeze to death. Again, because they also move. They can't stop swimming. And so they usually just keep swimming and can keep circulating oxygen that way. Great question. Thank you so much. Titus asks, how did you select the location where you collected the fish to be tagged? Really good question. So what I didn't go into in regards to the isotope study that I talked about was that a big part of that study was looking at where in Muskegon Lake the fish are sitting. And so they did a whole bunch of isotopic analysis, and you can use isotopes to kind of determine where a fish was recently in time and also in past in time. And so what they found out was that all the Lake Michigan migrants were coming in and kind of just going straight down to the bottom of Muskegon Lake where it's really cold. And so their isotope analysis, their those little like tags, I guess you would call them, let us know that they were using food and they were eating food from kind of the bottom parts of Lake Michigan. Whereas those residents were all kind of eating food that you would find along the near shore, so in the shallower parts. So when we were thinking about trying to tag fish, we kind of wanted to tag fish from both populations. And so we targeted fish in the bottom of Lake Michigan, and we also targeted fish on the outs, like in the not Lake Michigan, Muskegon Lake, sorry. So we targeted fish in the bottom of Muskegon Lake, and we also targeted fish on the near shore. Um, a big part of science is being able to work on your feet and think quickly. And so within a day or two, we kind of figured out that collecting fish from the bottom of Lake Muskegon Lake was not going to work. Well, no, sorry. We figured out that collecting fish near shore, Muskegon Lake, wasn't going to work for whatever reason. They just weren't biting. And so we pretty much had to take all of our fish from the bottom of Muskegon Lake. And that's kind of where that barotrauma came into play. So I hope I answered your question. That was a really great question. Kristen asked, did it matter if the fish were male or female in terms of who left the DLRMs, so the drowned river mouth lakes? We did not consider that. So I did take pictures of all their openings just to like kind of for our records, but it turned out that there really was our sample size was too small for us to be able to like definitively say yes, only males were leaving or only females were leaving. Um, that would be really cool to look at. We do have some students here looking at kind of furthering along this research. So we'll bring that up to them and I'll ask them. But no, the sex of the fish really, as far as I know, didn't really play too big of a role. Jamie asked what made you interested in aquatic science. So I had no interest in aquatic science until my senior year of high school when I took AP Environmental Science. And I just kind of had one of those magical teachers who just was super jazzed about science and jazzed about the earth and the environment. And she took a very like uh, pardon my language, but whole hog approach to teaching us, where she would kind of just put us out in the environment and ask us to collect like a couple things, but would basically just let us decide what we were interested in and kind of what we wanted to look at. And for me, what really got me excited about it was just the whole like hands-on part of stuff. So I really enjoyed being outside, stomping around, being in lakes, being in rivers. Um, and so I kind of just went with marine science. I could have gone with environmental science or fishery science, but those really weren't available to me, or I didn't know about them in time enough for me to make a decision. So my brain just kind of went marine science, and I just went with that. And I've been very, very happy with it so far. So all the pictures that you see on the screen are basically the reason why I do what I do, is because I love being outside, even if it's cold. So Anne asked, how long was battery life for the tags you used? Great question. So we used two different tags. We used a V7 tag and a V9 tag. We did that because we were a little bit concerned that we weren't going to get fish big enough to carry the V9 tags, which are a little bit heavier. So again, you need to make sure that your tag isn't affecting the fish's buoyancy to live. And so for the V9s, I think they were about 912 days. So we still have those tags coming in. And the V7s, unfortunately, they were done, I think at least 360 days after we started them. And so when you're thinking about science and you're thinking about starting a project, there's a lot of information that you need to kind of think about. And so that was a big one, is just like, are we willing to sacrifice a little bit of time to get just some data? And we ended up weighing the pros and cons and figuring out that, yeah, it was worth it to sacrifice some days to get data. Great question. Titus asked, Drawn Rivermouth lakes are a unique habitat to Lake Michigan, specifically to Michigan. Do you think your research will help understand other perch populations like Green Bay and Lake Erie? Yes, I do. So the genetic portion of my study is kind of a very small part of a bigger study that is in process right now. We used to have a research associate here who is building a genetic panel for which you will eventually be able to type your fish. So as you catch fish and you take genetic samples, you can, excuse me, log into this bank and be able to determine what fish your fish is. And where that's really important is that when agencies are doing their surveys of yellow perch, it's often unknown the origin of these fish. And so the more information that we can get about these fish, where they're moving, how they're moving, what time they're moving, what kind of fish they are, will help better understand how to put limits on fish for recreationals, for also commercials. For Lake Erie, I don't know if that I'm assuming at some point it'll kind of go forth to Lake Erie, but I do know that there are some Green Bay yellow perch within that study. Thank you for asking. It was a really great question. Anne asked, what was the estimated cost of the study and how is it funded? Super good question. Science ain't cheap. I'll start there. Science is not cheap. Each of those receivers that you see are about, gosh, maybe like at least $3,000. And we were generously given those. They were donated to us, which was huge. The batteries themselves are about 40, $50, and you have to change those every year. The tags, I think the total tag, all 40 tags, I think came out to like $40,000. And that's just material. That's just receiver and tag. So we had other costs. So we had boat gas, uh, car gas to get the boat to the lake. We have all the materials that you see on the screen. So I had to build that cradle that the fish is sitting in. That cost money. All the chemicals that we use, all the tagging equipment, so the sutures, the needles, the scalpel, all that kind of stuff. And then there was time, so our time costs money. And so I'm a graduate student, so it's not that huge, but other people we had to kind of pay to help get this put together. So an exact number I can't tell you because it's that would require me to do mental math, which I can't do right now, but it was very, very, very expensive. But acoustic telemetry it generally is pretty expensive in terms of money. Max asks, how often do you check the fish's location? Great question. So we opted for a two a year approach. Not a lot of people, but some people, depending on your question, will check more frequently because they really want to know like day to day where our fish moving. We did it once every 6 months, and that would require us to go out on the boat. If you could see on that top right picture, I'm holding a yellow cord. And so attached to each of my receiver pucks, so that main base is a yellow cord that then extends out. And then, so we have the GPS points for the main receiver and then for the anchor, which we attached a snag line. So that yellow line is a snag line. And so when we're pulling up receivers, we can kind of take a grappling hook and drag it in between those 2 points, pull up the receivers, take the data off, put them back down in the water. So we did that, I want to say 4 times because I've been here for 2 years, maybe 4 times. Yeah, just about. So, and that's what makes this project so interesting for me is that like I can collect and get all this data, but I only have to do a couple days worth of work in terms of like hard labor. So that was also really exciting for me. Great question. Max also asked, how does the genetics of your fish differ from humans? Really good question. Again, if I was a geneticist, I'd be able to tell you more about this. Um, Different. As you can imagine, the genetics that make humans humans give us hands and toes and hair, and what give the genetics that give fish scales, gills, and tails. So fairly different. I wish I could give you a more educated answer, but I really can't. Um, but I will send that paper to Jamie, and she can hopefully, if I think maybe you're a student, I don't know where you are, but that's the best I can do. Thank you for the question. My first question, I hope I wasn't able to answer. Kristen asks, if you could continue the specific study, what other questions would you want to explore? A phenomenal question. I kind of want to go more into where they're going once they leave Lake Michigan. I think it's kind of funny, and I think it would be really cool if we could see the yellow perch kind of hopping, like lake hopping. Um, but there's so many questions. Like we really don't know why these fish are moving into these systems. They're obviously not spawning. There's a master's student at Purdue who just started who I think will look at diets. Don't quote me on that, but I think he might look at diets. So that could tell us something. That's a really good question. Um, I wish our sample size was bigger because then we would be able to have a little bit more confidence in the fish leaving. Um, questions. I think that's it. I mean, I could ask questions until I'm blue in the face. I have plenty of questions up there. But yeah, thank you for that. Thanks for that answer for that question, Kristen. So I don't see any more questions. Feel free. Oh, we got one more. Titus asked, "Do you have more fieldwork planned?" I don't. So I am on the very tail end of my master's. I'm writing my thesis right now. So I actually don't have any more fieldwork planned, but I will say, um, if fieldwork is something that you're interested in, there are lots and lots and lots of agencies that hire seasonally. So a lot of summer work is available. So when I got out of my undergrad, I had zilch in terms of any sort of experience. I didn't really know how to even use a fishing rod or a fishing pole. And so I kind of got a job and I just jumped right in and they taught me basically everything that I need to know. So everything from how to put a trailer on a boat, how to put the boat on the trailer, how to drive the boat with the boat, how to drive the boat, how to drive the truck with the trailer and the boat, how to do do gas in the boat, how to do these like surveys that we do. So how to use different kinds of nets, how to hold the fish, how to collect samples from the fish. Um, and if that's stuff that you're really interested in, super good. Like there are tons and tons and tons of options in terms of like seasonal work that you can just get experience doing that kind of stuff. And that's where I fell in love with this field is that I was put in positions that I could kind of explore my interests in that field work. So kind of getting out and getting dirty. Um, so for this project, unfortunately, it is done. If we do have any more field work, it would be to go check those three receivers in the channel. But that might not be me. That might be somebody else because hopefully I'll be gone by then. Anne asked, what was the biggest struggle overall during the study? Great question. We had lots of little struggles. So I wouldn't say there was one big one. I would say there was lots of little ones. Mainly being the timing of this study. So I started my master's in fall of 2023, and my advisors wanted the receivers out, the fish tagged by November of 2023. So that was about 3 or 4 months of me just like really jumping right in, getting my hands dirty, calling everyone that I knew to how to do this. Again, not really a struggle, just like more circumstantial. It was just like I really got started working. I think one of the biggest struggles was that the fish up in in Charlevoix didn't seem to make it as well as we would have hoped. Again, we sent 20 tags up there, and we had 20 tags down here in Muskegon. And the ones that were up in Lake Charlevoix just didn't seem to work as well as we wanted to. So that was a bit of a struggle. We had some issues with the actual tagging of the fish. There was a little bit of a mortality problem, um, which is it is what it is when you work with fish. Some fish are very sensitive. So you look at a fish wrong, it's dead. Some fish are very hardy. You can kind of bang them around and not have to worry about them. These fish were a little bit in the middle where we were pulling them out of the water. They weren't looking great. We had to decide, is this fish viable? Can we work with it? Again, no harm, no like physical pain came to the fish except for being pulled out of the water by a hook. We anesthetized these fish so they didn't feel any pain. But we did have a couple fish not make it, and so we had to reevaluate how we were collecting our fish. That was pretty exciting. And then a big, just a normal struggle is science is hard. Science is hard. Science is complicated. We sometimes don't get the results that we would really love to have. But that is a huge part of being a scientist. Is just like you have these questions and in your back of your head you have these kind of expectations, and then those expectations may or may not be met, and you kind of have to do what you got to do with what you have. So that's been my struggle, is that like I really wanted this data to say something and it didn't, and I was upset about it, but that's just the way it works. Great question. Max says, do you ever want to be a professor of this kind of science? Yeah, so I've actually thought about teaching. To become a professor, I would have to go get a PhD, which I just don't have it in me right now. Maybe later on. I really would like to get out there and get working. There's two questions. There's that question, and Titus asks, what's next for me? A great question, both of you. I don't really have something concrete lined up. I am still kind of working on the writing process, but I have some things I would really enjoy doing. So I like doing stuff like this where I get to talk to people and explain what's going on. I have thought about teaching. I have thought about kind of working with agencies or schools or like nature centers and stuff like that to kind of bridge the gap between hard science and the community, trying to make it a little bit easier for the community to understand what the science is doing and saying and going, so that the community can make better informed decisions. I hope that answered your question. Anne asks, how do you/when will you retrieve the receivers? How do you ensure that they don't get lost? Super good question. So we retrieve the receivers in two ways. So those ones that are on the channel, again, they're attached to that chain. So those ones are pretty easy. We kind of take the boat, we drive it right up next to that emergency ladder. I reach out, grab it, and then for the receiver itself, there's a key, like a little dongle that comes with it, and you insert it into the receiver, and it's a Bluetooth. So that key will talk to your computer, and it'll offload that data via Bluetooth. It's really, really cool process. Um, For the ones in the lake and the ones in Muskegon Lake, we use a snag line. So I explained it a little bit earlier, but you have your main base, so the base that your receiver's on, and then you have an anchor, and attached in between these two is a snag line. And so when you're deploying these, you put a GPS point down when you drop the receiver, and you put a GPS point down when you drop your anchor. So when I went back 6 months later, I took a grapple hook, so just a really big hook, and I dragged it in between those 2 points. And back to the struggle question, that was a big struggle because we actually did lose a couple. So another part of science is just you lose stuff. You're kind of blindly putting stuff down into the lake and things get lost and you have to kind of adapt when those challenges come up. So there are, I think, 3 receivers that we were not able to recover. So if anyone's in Muskegon Lake and they find it, let me know because we would really love to get those back. Um, there was one that is completely lost. It got taken off the channel by wave action. However, somebody did find it, and there's a number on there you can call, and we were able to get that one back. So that was pretty exciting. Titus asks, doing research to answer questions just raises more questions. That's the fun part of science. No, it really is. It- Doing my master's, I get really into the weeds with things of just like there are too many questions and too many things I want to know. So it's a bit of a love-hate relationship right now, but I do still really enjoy being able to ask questions and then being afforded the opportunity to answer those questions. So getting to do all this field work and being able to go out there and make a dent in what is a big, very, very big proverbial science hill is pretty impressive and pretty cool. Max asks, can scientists do this on other types of fish? Absolutely. So that I very briefly talked about a organization called the Great Lakes Acoustic Telemetry Organization, and you're more than welcome to Google it. They have a website. And they are kind of the leaders in this acoustic telemetry stuff. So lots of people do acoustic telemetry projects on different kinds of fish. And then once they have that data collected, they can send it to this Great Lakes organization and they will kind of package it up and make it easier for you to understand. So I've had people I worked on a project when I worked in Alabama where I was tagging greater amberjack because we were trying to figure out how many there were and where they were going. I also was working on a project where I acoustically tagged sharks when I worked in Alabama as well. And that kind of gets into what tag for what fish because you can't put the same tag in every fish for a whole bunch of reasons. But yeah, you can tag anything. You can really tag anything and follow anything. You can put it on turtles, you could put it on squirrels. Um, a lot of people do this with birds as well. So you can, it wouldn't be acoustic telemetry, but just telemetry of birds. Um, it's a very versatile way to track fish, and it's a very cool way to track fish. Very shiny, very new, up and coming. It does come with its problems, but I think it's really cool. I just like the field work of it too. Any more questions?
So we don't have more open questions at this point. We have just a few minutes left in our time together. I'm going to go ahead and pull up. I'm going to ask you to stop sharing, Anna, if you would be so kind. And then I'm going to pull up a screenshot of one of our partner organizations that I was talking about before, the Center for Great Lakes Literacy. And let me just pull the slideshow up all the way from the beginning. This is the group that I was mentioning earlier called Seagull, and it's a group of educators across the Great Lakes Basin that promote a variety of different educational opportunities for both educators and students, including this one, Students Ask Scientists. You might want to check out the website. And I think Anne can either put that in the chat or the question and answer. It's very simple. It's cgll.org. A variety of resources on that website for you. If you would like to contact either myself, I'm Ginny Carlton, the Education Outreach Specialist with Wisconsin Sea Grant, or Ann Moser, who's our Senior Special Librarian and Education Coordinator here with Wisconsin Sea Grant. We'd love to hear from you with whatever additional questions you might have related to this topic or other resources that we can provide. So with that, I'm going to stop sharing and just open it up. We have a relatively small group today, so if you don't have anything else that you want to type in the Q&A, we can have you use the React button and raise your hand, and then I can give you permission to, um, speak to the group. Just recall that we are recording the presentation so that if you do speak, your voice is going to be recorded. So most folks are okay with that, but just want to make sure ahead of time. Kind of your last chance here.
So Ginny, I actually can't see anybody else. I can just see you and Anne. So if they raise
Yeah,
their
that's sort
hand,
of the
I can't
format
hear
of
them.
the webinar. Yep,
So if they do
yep,
ask questions, I just let me know.
yep, yep. So I don't have anyone raising their hands, so I think we're just gonna call it a day then. And thank you so much for your time, Anna. Thank you to your advisors for their support of
Oh
you
yeah,
and
big
all of
time.
the other work that's happening there at Purdue. And hope at one point maybe our paths will cross and we'll be able to meet face to face at some time.
Yeah.
Thank you to the educators for sponsoring their classes today. Obviously, we couldn't do this without the students, and thank you for helping to facilitate their questions and getting them typed into the Q&A on their behalf. We don't yet have another Students Ask Scientists presentation scheduled. We were hoping for one more this fall semester, but we may take a break until spring. Watch for announcements on the Center for Great Lakes Literacy website about those next offerings, and potentially add your name to our listservs if you'd like a direct email about them. Thanks again, everybody, and have a great rest of your Thursday.
Thank you so much.